So where we left off, we were talking about carrier-mediated transport, and I stopped and did some board work about the idea of a transport maximum. And we'll talk a little bit more about transport maximum later in the presentation, but for now, basically the idea is you have a finite number of these transporters, meaning that if there's so much of something in the filtrate that all of the sites on the transporter are occupied at all times, that's the concentration at which that thing will start showing up in the urine. So imagine you have so much glucose in the filtrate that all of the transporters available to transport it are always full. Any amount above that means that glucose is going to start showing up in your pee. So that's the idea of transport maximum. And it's clinically relevant because there are certain things like glucose that shouldn't be in the urine. So if they are there, that suggests a larger problem. In the case of glucose, specifically diabetes. So with regard to the regions of the nephron, we've got the proximal convoluted tubule, that's what PCT stands for, and the distal convoluted tubule. And then between them, the nephron loop. So the idea here, since we've already covered what goes on with filtration, the idea is specifically to take the filtrate that results and take useful things out of it, put those things back in the blood, and then move other things into the filtrate to get them out. So one thing that's not pictured in this image is the peritubular capillaries. So the efferent arteriole here actually branches back out to form a secondary capillary bed, which wraps all around these tubes. It's just easier to draw it this way so that you can actually see the loops. So just be aware that there are blood vessels surrounding all of the tubes you're seeing here. So the proximal convoluted tubule is kind of analogous to the jejunum. So the jejunum is going to be doing the most nutrient absorption. Likewise, the PCT is doing the most reabsorption from the filtrate. So anything useful that needs to be grabbed back from the filtrate is going to be mostly happening in the proximal convoluted tubule. So this includes ions, organic molecules like glucose, vitamins, so like water-soluble vitamins, specifically vitamin C, etc. And water follows along for the ride. So at the end of the digestive system lecture, we talked about how most of the water reabsorption is just passive due to absorption of nutrients. So water wants to be where the solutes are at. So if you're actively moving solutes into the blood, water will follow. Same concept applies here. The nephron loop is specifically for water reabsorption and sodium reabsorption. And it relies on something called countercurrent exchange, which is super critical for creating concentrated urine and allowing the urine to be at a higher osmolarity than the blood. So this helps keep control of body water osmolarity and body water amount, and we'll go through that. Distal convoluted tubule is for the reabsorption and secretion finishing touches. So anything that the PCT or nephron loop missed is taken care of here. And it's also an important place where drug and toxin metabolites are disposed of, so put into the urine. The collecting system, which includes all of the collecting ducts, the only thing that really happens here is water reabsorption. There are some solutes that come along for the ride, but most of what goes on here is water. And we'll talk about the effect of hormones on the collecting duct as well. Okay, so I mentioned that we were about to get into some conceptually challenging material, and this is where that starts. So I talked about transport maximum. And really what you're going to have to do to understand reabsorption and secretion, primarily reabsorption, is turn your brain back to Biology 160 and remind yourself of co-transport and antiport, and also concentration gradient kinetics. So the idea that if solutes are moving in a direction, that's going to tend to be the direction that water will follow, and the idea that you can use the concentration gradient of one particle to drive the transport of another. That's perhaps the trickiest thing to understand. So what we're looking at here, where I'm circling, is one of the cuboidal cells of the proximal convoluted tubule. The nucleus has been omitted just for clarity, and what we're looking at is the transport of a bunch of things from the tubular fluid into the cytoplasm and then into the blood. So each of these little transporters, so this one happens to be a co-transporter. Over here we have an antiporter. So antiporters, just remember, are when a protein transporter is moving two things in opposite directions. Co-transporters move two things in the same direction. Note, we don't have any ATP being spent here. So this process is passive, even though it may not look like it. This relies on a gradient set up by active transporters, and I'll explain that as well. So in order to understand the way this all works, I'm going to use my zoom in button. And we're going to focus first on this area. So this should look familiar in the form of the sodium-potassium pump. So this does use ATP. Unfortunately, the illustrator left that out. If it were up to me, I would include it because it's an important piece of information. So the sodium-potassium pump is an active transporter. I think perhaps they omitted it because they assume by now that that's common knowledge among students, but I feel that it's always good to be reminded which transporters are active and using ATP and which aren't. So I think that's an oversight. So just remind you, this is active transport right here. So there are more than one sodium-potassium pumps on this side of the cell. And there are many, 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 many per cell and many, many, many per proximal convoluted tubule. And they don't ever stop. They're always on. So they're constantly moving sodium from the intracellular fluid into the blood. So why is this important? Why do you need this constant flux of sodium? Well, if sodium is constantly being removed from the inside of the cell, that means that the concentration of sodium inside of the PCT cells is consistently very low. And in fact, it's consistently lower than the tubular fluid. So the reason why these transporters are always on is because it creates a situation where the concentration gradient between the tubular fluid and the cell fluid is very steep, with very low sodium here and very high sodium here. That creates a very steep sodium gradient. And of course, because of the way diffusion works, sodium is going to want to flow down its concentration gradient. And because the concentration gradient is quite steep, the energy stored, the potential energy there, is pretty high. So the same idea carries over for a hydroelectric dam, where the more water you have stored up behind the wall and the farther the water has to fall, the more energy you can generate using the dam. Same deal here. So this is a very steep sodium gradient, which represents a lot of potential energy that can be used to power the reabsorption of other things. So that's actually how this co-transporter works. So this doesn't spend any ATP, but it uses the steep sodium gradient to also successfully transport glucose and other organic molecules from the tubular fluid into the cell and then into the capillary. So it piggybacks on the sodium gradient and capitalizes on it to move something else. Similar deal with this sodium proton antiporter. So we've got a strong sodium gradient, and that is used to power the pumping of protons out. So there's consistently higher protons in the capillaries than there are in the cell. If we go down here and look at the arrows, you can see diffusion, which we know is passive, and then reabsorption and secretion. Unfortunately, they don't distinguish between active and passive in some places where it's key to understand that, but I'm going to fill those pieces in for you. So another consequence is we can see the carbonic acid buffer system present up here. So due to carbonic anhydrase activity up top here, we're going to have carbonic acid dissociating back into carbon dioxide and water, which is going to drive some of the osmotic water flow back into the blood. But we have those two things recombining here, and that actually keeps this proton moving in a loop. So in addition to the protons coming out of the blood, we also have ones dissociating from carbonic acid. And that proton loop powers this side of the exchange, so it keeps the protons flowing here. What that permits is for the sodium gradient to help move bicarbonate, HCO3-, into the blood plasma as well. Because remember, bicarbonate is useful as a buffer, but only if it's in the plasma where it can do its work, not elsewhere. Okay, so I would spend some time reading about this process and just sort of getting used to thinking about how the transport of one thing can drive the transport of another thing. Because odds are it's been a while since you thought about that, understandably. It's not something that's on normal people's minds every day. And so making sure that you're paying attention to it is a good idea for a full understanding of what happens in the PCT. So here's the outcome of that. We've got reabsorption of between 60 and 70% of the volume of filtrate produced. So remember when I was telling you about glomerular filtration and about 180 liters or so per day of filtrate produced, and I mentioned like, well, you sure don't pee out that much, so where does it go? Well, a lot of it goes back into the blood from the PCT. And remember, this is part of one nephron. We have millions of them. Oh, good question. So let's go back. So yeah, there are some other active pumps that are not included here, but they're part of renal physiology that we won't really get into. So for this particular picture, the only ATP expenditure that's really happening is here. Which is actually pretty convenient. But, and I'll think I'll show you this statistic in a couple slides from now, but if not, it bears mentioning that the caloric sort of budget for all of these pumps, because they're sitting there constantly spending ATP and your kidneys are constantly on, in a person with normal renal function, the sum total of all of the ATP expenditure from these pumps actually takes up about 6% of your daily energy budget, which is pretty crazy to think about just these little guys constantly doing their thing, taking up 6 whole % of whatever amount of calories you eat per day. So that's wild. So the reabsorbed solutes in water re-enter the peritubular capillaries, again, not pictured. And then the secreted solutes, like protons, for example, enter the filtrate and they become part of the urine. So when I was talking about the nephron loop earlier, I mentioned osmolarity. Of course, the nephron loop isn't the only part of the nephron that plays a role in osmolarity. Exchange between the urine and the filtrate and blood definitely contribute to that. So how much or little you're peeing and how much or little osmolarity that urine contains is a big deal. So remember, osmolarity refers to the number of solute particles per liter, and it doesn't matter what solute particles either. So when I say osmolarity and I express it in milliosmoles, what I'm saying is the bulk number of solutes, regardless of type, per unit body fluid, in this case, liter. So ordinarily, the osmolarity of body fluids is about 300 milliosmoles. We're going to see that change in the nephron loop and then change again once we get to the DCT. And thanks to the collecting duct, the osmolarity of the urine produced is customizable. So you can change it according to regulation needs for your total body osmolarity. So like, let's say if I'm too salty, if my body water is too solute rich, its osmolarity is rising, I can pee less and retain water and try and correct that problem. And the opposite is true as well. So if my body fluids are the wrong osmolarity in the other direction, I can urinate more to correct that. Okay, so PCT functions, in summary, I already sort of went through these, so this is just a summary slide. Reabsorption of organic nutrients. So these are going to include things like fatty acids, amino acids, monosaccharides, basically all the stuff we just talked about being absorbed by the intestines, because of course all of those nutrients, those monomers, are transported in the blood, which means that they're going to pass through the kidney, and odds are they're going to end up in the filtrate. So we want to get those back. Ions, including bicarbonate, and when it conjugates with carbon dioxide, of course, carbonic acid. And then we also have, of course, water, that's important, and the secretion of protons. So one thing that kidney is really good at is lowering blood acidity by scrubbing protons out. It's not especially great at holding on to them. So if you need to build up protons in your blood, your apneustic and pneumotaxic centers are going to change your breath rate and suppress inhalation and prevent you from hyperventilating so that carbon dioxide can build up in your blood and undergo the reverse of this reaction to produce protons. If you have too much protons, that's when you're going to start dumping more of them into your urine. So it's a sort of push-pull relationship between your respiratory system and your urinary system to accomplish a good balance of pH in your body fluids. And then, of course, the sodium role is really just, if you set up this really steep sodium gradient, you can use the sodium gradient as sort of a pack mule to carry other things, so to speak. Okay, renal threshold. I talked about this before, but it's a concept that's important, and it's also a concept that for a lot of students is counterintuitive, and I totally understand why. If you've never thought about this idea before, it's tricky. So I think it's fair to say that repeating it a couple of times is going to help with understanding. So saturated just means if you took a series of snapshots of all of the transporters in the membrane, all of them would be occupied at all times. So anything above that maximum, when all the transporter spots are occupied, is going to be the plasma concentration at which solutes will appear in the urine. So I gave the example of hyperglycemia resulting in glucosuria, and this brings me to a vocabulary note. So glucosuria means glucose in the urine. There are other kind of ureas. Generally speaking, if you see this suffix at the end of the word, it's going to be a medical term for the appearance of something or other in the urine. So for example, albuminuria is a condition where albumins from the blood show up in the urine. Typically that creates foamy urine. So if you use the restroom and then you turn around to flush and you notice like, wow, it looks like I just poured a beer, that's protein in the urine. It's not uncommon for people who are pregnant or people who are engaged in very intense exercise, so it's not a reason to freak out if you see it, but it's definitely not something that should be happening all the time. Okay, so nephron loop, also called the loop of Henle. Those two names are interchangeable. So for example, on the lab test, you can do either. And although technically Henle is supposed to be capitalized, if you don't capitalize it, I'm not going to count you wrong for spelling. So as long as everything is spelled right, I do not care. So this is the loop. And so in cortical nephrons, the loop doesn't really dip into the medulla very much, if at all. In juxtamedullary nephrons, the loop of Henle is quite a bit longer and it goes very deep into the renal medulla. And there's a significance to this, and that is related to the high osmolarity of the deep portions of the renal medulla. So before I explain that and why that matters, let's back up. The nephron loop does something pretty cool. It provides a means of concentrating urine such that you get a lot of salt and a lot of water back from the filtrate, and you set up this permanent gradient which exists in the kidneys to really efficiently pull things out. So let's look. And the first thing you need to understand this process is these two tubes are in close proximity. So in the illustration, they're artificially far away from each other, but in an actual nephron, they're pretty much right up against each other. So this little space here is really only occupied by a structure called the vasa recta, which is a ladder-like part of the paratubular capillary. So imagine a rope ladder, but it's made out of capillaries and it's intermingling with these two tubes. So that's what the vasa recta is. So that vasa recta is where the stuff that comes out of here is going to go. But the significance of the fact that the tubes are next to each other and that the fluid in those tubes is moving in opposite directions, that's really important. If the fluid in either tube were to stop moving, none of what I'm about to tell you would work. Okay, so here's our loop.
Notice we have two different regions, and they refer to lots of direction and sulfate traveling inside of them, so descending versus ascending, and also tubules and convoluted, so thin versus thick.
These also have different permeabilities. So the descending limb, when it gets thin, is permeable to water, but it is not permeable to solutes. So what this means is that increasingly high osmolarities of the interstitial spaces in the renal medulla are going to cause water to osmotically leave this portion of the tube. And again, it's not just going to leave and enter the medulla and equilibrate the osmolarity. It's going to leave and go into the surrounding capillaries. So this is a really efficient way to reclaim water from the filtrate. Now, the only reason this works is because in the ascending limb, we have sodium chloride transporters that are pulling sodium and chloride ions out of the filtrate and putting them in the renal medulla. So the renal medulla is very salty. So the movement of these sodium and chloride ions out is what creates this osmolarity gradient. So at the juncture between the cortex and the medulla down here, the cortex sits at about the same osmolarity as most body fluids, so about 300 milliosmoles. The deeper you go into the renal medulla, because of this sodium transport, you get 600 and then 900 and then 1200 milliosmoles at the very deepest parts of the renal medulla. So 1200 milliosmoles is high, and that's going to cause water to really want to move towards that 1200 milliosmoles. So the active transport of sodium and chloride out of the thick ascending limb, and the fact that this portion is impermeable to water, creates the driving force that pulls water out of the descending limb and puts that water back into the blood. It's also critical that these two fluids, so the fluid that's losing water in this limb and the fluid that's losing sodium chloride in this limb, are both constantly in motion and both constantly undergoing transport, either of sodium chloride or of water. So this removes 2/3 of the sodium chloride that enters the loop. And if you take a moment to reflect on the importance of these ions, so for example, you need sodium to do action potentials, right? And those are critical for brain and neural function and also for muscular function. And then if you think about chloride, you need this for a variety of processes, perhaps the most important one being the chloride shift. So that's how you get bicarbonate into and out of your red blood cells. So it makes sense that we would want to reclaim these ions anyway. And as we do, we also have the added bonus of using that concentration gradient to reclaim some water as well. So the nephron loop is exciting to physiologists, and indeed to me, because from a design perspective, and I'm not saying like I personally believe in intelligent design, that's, you know, this is not the space to discuss that, but rather just like if I was going to sit down and try and design a water reclamation and salt system, I couldn't design anything more elegant than this. This is very impressive in its simplicity and its effectiveness. So the pumping in sodium and chloride ions out basically makes the gradient I mentioned. And this process is going to continue as long as we have good GFR. So one of the consequences of a drop in GFR is that the motion of this fluid declines, which starts to change the osmolarity gradient. So that can impair renal function and also impair the ability to regulate body osmolarity. So this is one of the reasons why hypovolemic shock, so shock due to low blood volume, is very, very dangerous and ends up causing organ failure. The kidneys are often the first to go in that situation. And because they're so important in regulating things like body osmolarity and ion concentration, other organs begin to follow shortly thereafter. So really, the kidneys are sort of the crux of survival in situations like that. So this is why medical efforts are made to preserve the kidneys. There's sort of an order of operations there, and kidneys are pretty high on that list. So another consequence of that concentration gradient set up by the nephron loop. So pay attention now to, here's our nephron loop. So we know we go from 300 up to 1200 and then back to 300. And then by the time we get to the DCT, because of continued removal of things, now we're down to about 100. So if you were looking at this and thinking, I thought you said it was for concentrating the urine, but we're going back to the original osmolarity. So this filtrate doesn't seem especially concentrated to me, and this especially doesn't. So if you're scratching your head about that, you're right. That's confusing. The nephron loop sets up the gradient and the preconditions for pulling water out of the collecting duct. So the place where the urine is capable of being concentrated is here. The place that sets up that possibility is the loop. So again, we have a tube with fluid moving in it in close proximity to a set of tubes that are manipulating osmolarities. That's the significance. So if I know that there's an osmolarity gradient where at the area where the cortex and the medulla meet, things are about the same as regular body fluid, and then the further down in the medulla I go, the higher the saltiness factor is, so the higher the osmolarity. That means that even if this filtrate is sitting here at 100 milliosmoles, as it descends the collecting duct, which passes through the medulla to get out, the collecting ducts exit at the renal papilla, those ducts pass through this increasingly salty zone, meaning that the further down you go, the stronger the osmotic pressure is to move water out of the collecting duct and into the medulla. So if you look, as you descend in the collecting duct, 300, now we have water that wants to move towards 6. And that's going to increase to 6-ish, and that water wants to move towards the 9, and so on and so forth until you end up with urine that is a concentration that's about the same as the renal medulla by the time you get to the exit. So again, the tube sets up the precondition for reclaiming water. The collecting duct actually makes that happen. So you need all 3 tubes, descending limb, ascending limb, and collecting duct, to effectively concentrate urine. So the name for this is countercurrent multiplication. So countercurrent refers to the fact that the fluids are moving in opposite directions. And multiplier means there's a multiplicative effect on the changes in osmolarity. So the power here is the more permeable you make the collecting duct, and later the papillary duct, the more water will leave. And in fact, there is a hormone that changes the permeability of this and makes it more permeable. We'll talk about it later. But for now, we got to talk about the distal convoluted tubule. So not to give it short shrift or ignore it, it puts the finishing touches after the nephron loop situation is completed. And it does some stuff as well. So it's going to do some finishing reabsorption and secretion, anything that was missed by the PCT. Also, unlike the PCT, it is sensitive to hormones, specifically parathyroid hormone and aldosterone. So remember, aldosterone is going to act on the sodium-potassium pump to change its action and selectively hold on to more sodium at the expense of more potassium. And then, of course, parathyroid hormone is going to change the way that the kidney deals with calcium ions. So it's going to cause the kidney to retain calcium and move it into the blood at higher rates than it would if PTH was not circulating. So I believe I first introduced students to parathyroid hormone all the way back early in 241 in chapter 6 when we discussed calcium homeostasis in the context of skeletal tissue. And now we're circling back to exactly how it performs that function in the kidneys. So everything is connected all of the time. Unfortunately, that's why learning this stuff is so difficult, but also the human body is really amazing. All right, so here we have some more co-transport. And again, unsurprisingly, we've got a sodium-potassium exchange pump setting up this sodium gradient and pulling whatever sodium is left after the nephron loop out of the filtrate. So we have co-transport of sodium and chloride, and potassium is the expenditure. So we're going to move potassium into the tubular fluid and the urine. Fortunately for human beings, potassium is not really what we call a limiting nutrient. So a limiting nutrient is one which is rare in the environment and therefore hard to get. Potassium is not one of those. Potassium is super easy to come by. Almost everything has it. So the moral of that story is that we can afford to use it as a bargaining chip to get more sodium and to set up sodium gradients. So if aldosterone is present, we're going to not only have sodium-potassium exchange happening at the basal surface of the DCT cells, but also at the apical surface. So effectively, this is going to double or even more than that, the amount of sodium being reclaimed. And that creates additional driving force for water to come along with it and re-enter the blood. So that's really the way aldosterone works, is it creates extra sodium suction across this interface here, and water will follow. Not pictured in this picture, but also true, is if you were to see parathyroid hormone circulating, you would see more transport of calcium as well. So here is the picture of just aldosterone-mediated potassium secretion. This is great, except for one consequence of this: if aldosterone is released chronically, we did talk about this in the endocrine system in the form of the stress response. Consequence of chronic release of aldosterone is a syndrome or medical condition called aldosteronism. And aldosteronism creates a situation in which you end up with hypokalemia, meaning you have abnormally low levels of potassium in your plasma. And even though potassium is not a limiting nutrient and it's relatively easy to come by, if you're starving, that's not really true. And potassium does have a really strong consequence on the membrane permeability of things like neurons and muscles. So you end up with nervous system excitability changes that can be really, really bad for survival. So aldosterone is like a Goldilocks hormone. You want not too much, not too little, just the right amount. So distal convoluted tubule. Here's another place where you're doing additional secretion of protons. So here we have another exchange pump. And in this case, the exchange pump is not sodium-potassium, it is sodium-proton. So again, we're setting up a driving force using active transport of sodium, but in this case, instead of exchanging it for potassium, we're exchanging it for a different cation, which is a proton. So this is going to help to move protons out of the blood and lower the acidity of the blood, so increase the blood's pH. So there's two routes for this. There's diffusion, driven by concentration gradients that are set up, and you can also use something called deamination. So amino acids, as you know, have a central carbon, and then they have an amino group, NH2, right? And a carboxyl group, COOH minus, on that end that's the same for all of them, not the R group. Deamination means pop the amino group off of the amino acid. So take that NH2 away. And that results in the production of protons. They also both generate bicarbonate ions that are returned to the blood. So the benefit of performing this, where we have, here's the product of deamination. So you get NH2 being removed, and then some more protons jump on, creating NH4. And those are moved into the tubular fluid, creating ammonium chloride. And the remainder turns into bicarbonate and goes into the blood. Sodium also is entering the blood, and those actually combine to form sodium bicarbonate, which is another part of the blood buffer system, one that we haven't discussed yet. So if there is lots of sodium in the blood, it tends to naturally combine with bicarbonate. Oh, wait, hold on. Before we move on, I wanted to point out that this should look familiar. So the exchange of a chloride ion for a bicarbonate, putting chloride where it's not needed in exchange for putting bicarbonate where it is. The place that you have seen this before is in red blood cells, while the red blood cells are exchanging gases with metabolically active cells and tissues. So chloride shift does happen between red blood cells and your plasma, but it's also useful in other places. It just doesn't have the same name. So this is another place where chloride is kind of used as a bargaining chip to move bicarbonate around. So the collecting system I mentioned is sensitive to a hormone. And technically the DCT is included in the collecting system, primarily because it too is changeable in its permeability due to the action of hormones. So aldosterone primarily acts on the distal convoluted tubule, as does PTH. Antidiuretic hormone primarily acts on the collecting duct, specifically to put these proteins called aquaporins into the duct. So aquaporins are just holes through which water can move. And remember, we've got 300 milliosmoles up here, 600, 900, 1200. So if we put more holes for water to pass in this tube, more water will pass out, and that helps to concentrate the urine. So ADH causes you to urinate small amounts of very, very concentrated urine by putting extra water holes in the collecting duct. It also creates reabsorption. For example, the collecting duct, as it approaches the renal papilla, we call it the papillary duct. So same tube, just changes names as it's closer to the tip of a renal pyramid. And those are also permeable to urea. So that ammonium chloride that is entering the filtrate, soon to be urine, a lot of that gets turned into urea because ammonia and its chemical relatives are more toxic. So we get the production of urea, and then urea moves back out into here. And that contributes to that concentration gradient as well. So secretion regulates the pH of body fluid. So if the pH of the peritubular fluid is low, we secrete protons. So get protons out of our fluids and into the pee to pee them out. And if the pH is high, we scrub bicarbonate out instead. So let me see where we're at here. So this seems like a good place for a short break because we're about to switch topics a little bit. So let's take about 2 or 3 minutes to use the restroom or do whatever you need to do, and then we'll rejoin and finish this up.